So thank you very much, Marlon, for introduction, and thank you for inviting me back. It's always a great pleasure to see old friends and both, faces that I'm familiar with and the new faces, and of course all of his, great new buildings and all of his great things, and most importantly, great science, which is still taking place here. So what I'd like to do today is to talk about a couple of new research directions, which we actually started over the last few years, and they're now bearing fruit. So specifically what I want to do is to talk about two kind of experimental efforts, one involving, I would say, mostly quantum optics type physics, but which involves the interface between, let's say, atomic physics, physics of ultracold atoms, and physics of solid-state devices, in particular solid-state photonic devices. And the second part of my talk, and this is a work which we're doing in close collaboration with Vladimir Vuletich's group at MIT. And at the second part of my talk, I will switch gears, and I will talk about our effort involving using the quantum systems as very precise and very unusual sensors. And this will be a part involving diamond sensors. And this part actually overlaps with some ongoing work in condensed matter physics, but goes all the way down to a biological physics. And I think this maybe is appropriate given the kind of a huge bandwidth of the kinds of things you guys are doing here. But first, I want to mention that all of this work is a quest, is a part of ongoing effort which essentially involves a quest for controlling quantum world. So in this quest, what we are trying to do, we are trying to isolate and control some simple quantum objects, like individual atoms, individual spins. And what we try to do with them is once we gain a control, once we can, for example, cool them, once we can prepare a well-defined quantum states, we then use them as kind of building blocks to build more and more complex systems. And there are a couple of things which we are exploring with this. One of them involves the new physics, which we're exploring with some kind of synthetic or engineered systems, and in particular looking for things like new states of matter, or possibly in exploring some new applications. And for example, most of you probably have heard about quantum computing and quantum information science. This is of course part of this field. So here is kind of example physical systems which represent a kind of playground, the systems which we are playing with. So photons, for example, are playing an important role in this field because they are very important for communicating information, both classical and quantum, over long distances. And basically, you cannot really beat the photon as a carrier of information, but the problem is that photons generically do not interact with each other, and it's also kind of hard to localize and store and, for a long period of time. At the same time, isolated ultra-cold trapped atoms and ions have emerged as leading candidates in this field of quantum science, and the reason for that is that these systems, which are essentially particles suspended in vacuum tubes, have excellent isolation. So they are among the most coherent, the best coherent systems available, and you can control this coherent states of these individual atoms and ions with very high precisions using, for example, lasers. However, the problem with those systems is because of their isolation, they interact very weakly to each other, and they're hard to, for example, to envision, some large-scale integrated quantum devices based on this essentially kind of vacuum tube type technology. In the last few years, these ideas of quantum control have penetrated quite deeply into the field of the condensed matter physics, and there is now a wide variety of solid-state quantum systems which are being explored, such as, for example, quantum dots or superconducting quantum bits. So these systems are, stable in place, and they have very good potential for integration. And because they are localized, often to nanoscale dimensions, they feature strong interactions. But those systems live in complex solid-state environment. And for this reason, generally, their coherence properties are not as good as those of atoms or even photons, and they are sort of hard to control. So this kind of shows you that while there is a lot of interest in this field, there is really no kind of single candidate system which emerged, which is basically perfect, which combines all sorts of desirable properties in a single setting. And what is the key challenges in this field is that basically what you'd like to do, you'd like to essentially isolate and control system composed of many individual quantum systems. So this is, for example, this challenge of isolation and control of many body systems. So there are many experiments where 1 or 2 or 3 quantum bits are isolated, but it's really not clear how to basically keep increasing the size while at the same time really maintaining this very good degree of control. At the same time, as I just mentioned, we do have systems in the lab which we can prepare and manipulate. They are generally small systems involving few quantum bits. And then, of course, one could ask a question, given this kind of system, can we do some interesting science, or can we use them for some kind of application? So that's a challenge, another challenge of application of small-scale quantum systems. So what I'd like to really focus on this talk is the idea of exploring this some kind of hybrid approach, where I would like to kind of take advantage of coherence associated with isolated atoms, or maybe atom-like systems, and of strong interactions which are associated, for example, with nanoscale solid-state systems. And the aim of this research is basically try to combine the best of all worlds. So we would like to use with very good coherence associated with atoms and strong interactions, integration, scalability of the solid-state devices. Just to give you a little bit more specific examples of what we would like to achieve is that, for example, with this type of approach, what we would like to do is we would like to couple atoms directly with tightly localized photons or phonons, and that, for example, might allow us to use atoms as means to store quantum information, and use these other degrees of freedom as means to communicate quantum information. This could be, for example, a new approaches to quantum processing and communication. At the same time, as we learn how to control and measure kind of atoms or atom-like systems at the submicron scales, this by itself really opens a door for some new remarkable possibilities. And in particular, one possibility is to explore these quantum systems, in particular explore the fact that the quantum states are fragile for sensing and metrology applications. And the kind of the key thing is that if you start with this with the sensor, which is very small, you could push this kind of metrology all the way down to kind of nanometer length scale, or maybe even beyond. Now, these are all remarkable possibilities, but of course some of these things are challenging, and the challenge is really to try to isolate coherent atoms, individual atoms or atom-like objects which are near or inside nanoscale solid-state systems. So it actually turns out to be a very hard problem because it involves basically the interface between, let's say, quantum optics, material science, surface physics. But oftentimes these are the kinds of problems which are the most fun, and these are these efforts which now really span physics, photonics, chemistry, nanoscience, and actually go all the way down to biology. So within this kind of broad field, what I'd like to do is to give two examples of our recent work in this area. So one of them involves what I'm going to call a nanophotonic interface for ultracold atoms. So basically, I will show you how we can make systems of the kind shown here, where basically this is like a tapered a fiber tip, where to the end of that we attach a very thin, tiny kind of a nanophotonic device. In this case, it's just a dielectric kind of waveguide with some holes. And what we can do, we can take single individual ultracold atoms from the gas and bring them right in direct proximity of this type of device. And with this kind of system, we are now starting already to explore some interesting applications. For example, I will describe you an application in which we build a single photon switch. So basically, this is like kind of a light switch, which is activated by just individual light quanta. So when the single photon comes, basically turns on the light or on or off the light in a room. That's kind of a simple thing to think of. So in the second part of my talk, I will focus on a different types of physical system and a different type of application. So I'll focus on our efforts to extend the magnetic resonance imaging technique all the way down to individual molecules and sensitivity all the way down to single spin or single atom. And this approach will make use of atom-like solid-state defects as nanoscale sensors. Okay, so first, let me kind of motivate our work on kind of developing the interface between single atoms and single photons. So this kind of process where just one atom interacts with one photon is a long-standing kind of problem in optical physics. So it's, of course, one of the most fundamental process in light-matter interaction, but also the ability to really, engineer and make this process, work in a coherent way have many potential applications in both quantum and actually classical information processing. So, for example, this type of process can be used for to connect small quantum computers together into the kind of quantum internet. So where the idea is that basically you store the information, for example, in individual atoms, and you build some kind of small-scale quantum register on quantum computer. But when you want to couple this quantum register to another one, you just basically map the information stored in the atom in the photon, and it will go to the other kind of memory node, and then you can basically, that way enable communication between them. So another example is what I try to illustrate here, but maybe because the kind of projector decided it's such a futuristic device, it does not really show it. So this is kind of the example where you have a photon switch, which basically controls the flow of light, which in principle can be activated by just a single individual photon. So this is kind of an ultimate limit of nonlinear optics, where one photon, one quantum of optical radiation field can come and basically turn the switch on and off. So these ideas have been around for a long time, but it's actually very challenging to implement. And the reason is that if you generally send one photon and kind of try to focus it on single atom, the probability of interaction will be actually quite small. And moreover, photons can be easily lost. For example, they can be scattered in the modes other than the modes that you can control. So for this reason, in general, single optical photons, I would say, are not under the same degree of control at the moment as compared to, for example, single atoms, single ions, or maybe single spins. So in what follows, I'll describe a new approach to this problem. But before I delve into kind of details, I would like to talk a little bit about the physics of single atom, single photon interface. So let's consider a situation where you just have individual atom, and we all know if you excite this atom, it will decay via spontaneous emission with the radiation, which will have some kind of dipolar pattern. So in general, it will decay kind of in all possible directions. So what, how can I try to, modify this process? So one way to do that is to put a cavity around this atom. And so what this cavity will do, it will actually enhance the interaction of this atom with a certain set of modes, with the modes which are confined by this cavity. So in general, the way to quantify this is through basically coupling of single photon in this cavity to the single atom, which will have some characteristic Rabi frequency, which will be proportional to the basically a dipole moment of the atom and the electric field in the cavity per photon. And it's very clear that if you, for example, squeeze the cavity, if you make this resonator smaller and smaller, then the energy density is just basically due to the shrinking volume, the energy or the electric field per photon will grow. And so what this means is that, if you make the cavity, sufficiently small and sufficiently good, what can happen is that basically at some point this interaction with the cavity mode will start playing an important role, and then instead of just emitting in a free space, this atom can actually emit a photon in a cavity mode. So once you emit a photon in a cavity mode, of course, this photon will eventually kind of leak out of this cavity. This leakage rate is given by the rate couple. Basically, is determined how good are these mirrors. So basically, the figure of merit for this kind of single atom, single photon interaction is often called cooperativity, which essentially is the ratio of this decay of the atom into the cavity mode versus a free space decay. So basically, this cooperativity determines how much the cavity modifies the interaction of light with matter. And so this cooperativity can be expressed in terms of these parameters. For example, this single photon kind of Rabi frequency and this cavity decay. And actually, if you just use this type of consideration that with shrinking mode volume, this G grows. So what you find is that cooperativity is fundamentally determined by two parameters. One of them is a quality factor of the cavity, and another one is mode volume. So conventionally, the kinds of experiments which I will talk about essentially involved starting with the cavity, which is pretty sizable. But then what you try to do is just try to make mirrors better and better, increase the Q. So, however, in what follows, I will describe a new approach in which I will enter the so-called Purcell regime, which is named after Purcell, which corresponds to the situation when basically you excite this atom, it will mostly decay into the cavity mode, so the C much larger than unity, but this will be achieved by playing with this V, by shrinking V as much as possible. So in particular, I would like to explore the ideas of subwavelength light localization to basically create the cavities with mode volume on the order of smaller than lambda cube. So that's essentially the idea of this approach. So we will start with the light, which is localized to subwavelength dimension in a certain type of photonic structure. And the kind of photonic structure which we will be using is a so-called one-dimensional photonic crystal. So essentially, what it is a kind of a little tiny dielectric wire which can guide the light. And in this wire, what we'll do, we'll just put in, drill some little holes. So these little holes can, basically create a band gap for light. So this case senses behaves as a one-dimensional crystal for light, one-dimensional photonic. It will be made from silicon nitride. It's a dielectric material with low loss. And the idea will be to somehow take atoms and then place them very close to this kind of wire. And so, if these atoms are in the vicinity of the wire such that a evanescent field of the guided light can talk to the atoms, then this is what we achieve the desired result. So, but this kind of sounds simple, but it actually turns out to be quite challenging because basically what you need to do, you need to figure out how to take individual isolated cold atoms and bring them and keep them, in the proximity to this device. So the way, and so it's actually been like a long-standing effort to try to do something like this. And actually, a few years ago, we embarked upon this task with my friend and colleague from MIT, Vladan Ubiquitin. So the idea to how to achieve it is actually turns out to be very simple. So what we do is we start with something which is called an optical tweezer, which is basically a focused beam of light. So if you focus a beam of light, then this focused beam of light creates basically a potential for the atom, right? So the atom can be attracted to the high intensity part of this beam. And what we do then is we just take this optical tweezer and shine it into this nanoscale solid state system of interest. And what will happen in this case is that almost all systems will reflect a little bit of light. And this reflection of this light will create kind of a, standing, wave. It will create intensity modulation here on this half of the space. And what will happen is that basically they will create a kind of periodic potential for the atom in direction perpendicular, transverse to this structure of interest. So what we can do then, we can use this newly created modulation as a kind of optical lattice. So essentially, what we'd like to do is we'd like to put the atoms right in this point, which is basically the first intensity maximum, right, after the reflection from this system of interest. So, so if we can achieve it, then basically we achieve our goal. So the atom will be sitting right next to this nanoscale solid-state system, and then basically interact with the photons which will be guided through the system. So this method and this idea sounds to be quite simple, and actually this method is, in principle, very general. But to implement this, it took a lot of actually hard work. And in particular, these are the two individuals. There's a graduate student actually quite amazing individual, Jeff Thompson, and one of the postdocs in my group who really made this to work. So here is, some details about what is exactly we are, the kind of system which we are using. So we start with the wires made from silicon nitride. This is just a dielectric, low-loss dielectric, and then we drill these holes. So these holes act as like broad mirrors. And in order to create a cavity, what we do, we make more holes, but we just modify the hole spacing a little bit. So it creates like a defect in the crystal lattice. And this defect can maintain, basically corresponds to a cavity mode, which has the mode volume, effective mode volume, which is actually subwavelength, which is about half of the lambda cube. Most remarkably, we can, by sort of engineering these holes in the right and appropriate manner, we can create a cavity here, which is actually has fairly high Q. So in fact, in a kind of most recent work, we created cavities which has a Q of 0.5 million, so which is probably the highest cavities visible of this kind which has been created. So in this work, which I will tell you about, we basically use the cavities which with lower Qs of about 15,000. But what's also important is that these cavities can be radiatively damped. In other words, the light which is confined in this cavity can be directed outside into this waveguide, and from this waveguide we can actually extract it by using an optical fiber, a tapered optical fiber, very high efficiency. And then at the end of it's optical, it's all in optical domain, so it will be about around 800 nanometers, right? So it's all in the optical domain. So what we'll do is then we'll just bring the atom, in a tweezer and put it right next to it. So here is actually how this is done in practice. So basically, we mount this photonic crystal at the end of a tapered fiber in the vacuum chamber, and then we start with a focused beam of light where we grab individual atoms in this tweezer. Then we cool these atoms to near the ground state, and then just by moving the mirror, we move this optical tweezer to overlap with this photonic crystal. What's illustration shown? This illustration basically shows what happens with the light intensity. So what we see is that when this light, when this light beam, is pointed at this photonic crystal, we observe that there is this kind of first intensity maximum is created. And basically, if you start with the atom which is sufficiently cold and properly positioned, we can just bring it all the way to this first intensity maximum of nearly 100% probability. So that's how we create this system, which basically a solid state, photonic crystal, and a single atom. So the length scale here is about 200 nanometers, but the size, but of course the atom is actually pretty cold. So the atomic, the size of atomic wave function is, maybe 20, 30 nanometers. So it's actually it's close to the ground. So it's basically it's a harmonic potential, and the atom sits near the bottom. So this is at the end how the system looks like. So this is a photonic crystal fiber. This is a fiber, and this is this photonic crystal attached. So this thing, actually, we use it as a heater. So we tune the resonance frequency of the cavity by heating this structure, so which actually results in quite an amazing system, because this atom is cooled to something like microkelvin, while the temperature of this photonic crystal we estimate to be something like 800 degrees. So it actually gets very hot, and it really has very little consequence somehow. But so what we can do then, once we position this atom, we can just excite it and look how it emits. And basically what we observe is, just observe spontaneous emission. But note the time scale that this atom here basically loses its excitation. It's excitation on the time scale of maybe 3 nanoseconds or so, which is actually much faster than free atom decay. So that is really the signature of this so-called Purcell effect. So the cavity modifies the emission from this atom such that it basically speeds it up, so because of presence of this localized light. And this speed up depends on atom cavity detuning, so it's maximal when the atom is tuned right on resonance with the cavity. And actually from this kind of, enhancement ratio, from this cooperativity of 10, we can actually estimate what are these cavity parameters. And what's most remarkable is that this quantity G, this single atom Rabi frequency, single atom, single photon Rabi frequency is actually about a gigahertz. This is lifetime. This is a lifetime in the cavity. This is basically the spontaneous emission. This is this is like an outcoming pulse, the fluorescence in the pulse as a function of time after the excitation. And this is the enhancement of the emission rate as a function of atom cavity detuning. I'm sorry that somehow this projector it doesn't show very well, right? So basically, we enhance spontaneous emission by a factor of 10. So that's actually kind of an interesting. That's an interesting question. Maybe there is a little bit of real beating, and it might be a consequence of atom moving a little bit inside the trap, so that it basically feels so. But of course, this also note that this, at this point, the atom atomic excitation is, decayed quite a bit, so it's kind of not completely clear. Okay, so now we have assembled our system. So I just want to kind of finish this part of the talk with one kind of little simple kind of cute physics experiment which we have done. And this basically this experiment involves basically a realization of this single photon switch, this kind of device where one photon comes and turns the switch on and off. The idea is actually quite simple. So let's first consider the light propagating, light propagating in some kind of waveguide where we unfortunately don't see this waveguide. But imagine, think about this as basically photons flowing, in the pipe. And so imagine that we have one atom which is sitting in this pipe, and imagine that this atom is kind of strongly coupled. So what this really means is that this atom essentially will block the flow of photons in the pipe. And so what will happen in this case? Well, because the light is confined to one direction, it actually can only go back. Right? It hits it. It's like, you plug the pipe, the photon, hits and goes back. But, but think now about this. So this is just one atom, right? So what this really means is that if we have, instead of one photon, a photon pair coming here, then what can happen is that the first photon can actually saturate this atom, right? And then for this kind of photon which comes with that, it will basically, its flow will no longer be blocked. So what if two photons come? No, so it's two photons which come very close to each other in time. So it's two photons which come to the atom within the atomic lifetime, right? So if photons come one by one, right, they just, come and, reflected everything. But if photons come, very closely, spaced closely in time, then what can happen is that one of the photons, it's a kind of hand-waving picture, but one of the photons can excite this atom. Then the second photon. No, they are actually coherent. It's actually are coherent. So what happens in this case is that essentially when two photons come and interact with this one atom, they are much more likely to be transmitted in pair. So effectively, what happens is single photons will be reflected, but photon pairs are much more likely to be transmitted. So actually, of course, I'm kind of now. So I'm speaking now in a simple analogy. So actually, what happens? The real physics. What happens is that you end up with atom-photon bound state. Exactly, and this atom-photon bound state then tends to. So all of these interactions are coherent, right? So all of this it's a one-dimensional. It's a 1D kind of bosonic channel with a two-level impurity, right? So it's actually related. It has a lot of relationship to, like, mathematically, it's connected to things like Kondo model and things like this. So it's actually it's an interesting problem. Yeah. So, but basically, what will happen then? Single photons will be reflected, and photon pairs will somehow be preferentially transmitted. So what you see now is that atom, in this case, kind of acts as a single photon switch. But moreover, if somehow we can then add something to this atom that it will remember about whether the first photon comes or not, then you can actually, if you add the memory, you can convert it into something like a transistor. And so this will be this kind of device which I tried to illustrate in the beginning. So basically, one photon will come and turn this on and off, and then this will control the propagation of the subsequent photons. So if you add the memory. But okay, so let's first try to see if this simple picture actually holds in the experiment. So the way how we'll do it, we will use our atom cavity system. So in our case, what we'll do, we have essentially asymmetric situation where we, on this photonic crystal, we make many more holes on one side than another. So what essentially this means is that one of these mirrors will be perfect and another one will be partially transmitting. So in this case, the photons, can really never be lost. So when the photon comes and hits this cavity, it will be reflected. If we put an atom in this cavity, it will also be reflected. But actually what will happen is that the presence of the atom will imprint a phase shift on this photon. And of course, if we then put this kind of system into the, into the interferometer, then we can basically make complete analogy to what I described before. So just one of these ports will be kind of a reflection port, what I described before, and another one will be transmission port. So what will happen, once again, the photon will come, will interact with this atom in the cavity and get some kind of phase shift, and then depending on the phase shift, it will either go back, corresponding to reflection, or will go up, corresponding to transmission. And basically, in a system like this, what we measure is precisely this kind of what I predicted on the previous view graph. So basically what is shown here, and I again apologize, one does not see this, the axis. So this is a second order photon-photon correlation function. So basically what we measure is, conditioned on that you observe one photon, what's the probability that the second photon comes within a short, time interval. So in this reflection port, you see that these photons are completely anti-bunched. So that means that photons only arrive one at a time. But in this transmission port, what you see is actually bunching, right? So this really corresponds to this kind of photon pair. So, and what you see, kind of, if you take a step back, what you realize here is that clearly this shows that the system is nonlinear at 2 photon level. So this kind of atom as a kind of acts as a quantum sorter. So it basically it sorts one single photons and photon pairs, it sends them different way, in a different direction. So, but remember, I promised you that, I will kind of describe this, this photon switch. So in order to implement photon switch, you have to add memory. So you can add memory by actually just adding additional internal state to this atom. So this could be, for example, 2 hyperfine states, in our case, 2 hyperfine states for rubidium. And so what we'll do to realize the switch, we will prepare atom in a coherent superposition of these 2 states. Now, when photon comes, it will only interact with atom if it's in one state, in the state 2. The state 1 is far detuned from interaction. And remember what I told you, when the photon hits this cavity, it kind of exhibits a phase shift depending on whether atom is present or not. So this is a phase shift imprinted on the photon. But of course, in the single atom, single photon interactions, these are really elementary quantum mechanical interactions, things must be reciprocal. So what it simply means is that when photon comes and hits this kind of cavity, upon reflection, there is a phase shift, but you can think about this as a phase shift on a photon, or alternatively, you can think about this as a phase shift on this atomic superposition. So what this means is that basically when one photon comes, it hits this cavity, it will actually modify the phase of this prepared atomic superposition. So you can then think about kind of a simple idea for this quantum switch, and the idea would be to essentially do a kind of generalized Ramsey experiment. So Ramsey experiment, what it is involves preparation of the superposition of these two states. So then you wait, then you send a photon or not, and then afterwards you basically convert the superposition into populations, and then what you could do then, depending on whether the atom eventually is in a state 2 or 1, it will either basically reflect the photons with the phase shift or without. And that, of course, you can convert easily into basically light beam going one way or another. So that's precisely this kind of this kind of idea. The one photon comes and switches the light on or off. So it's actually in our case it switches the phase of the light. The phase is either has one value or another. And so this is kind of the summary of the experimental results. So basically what we do is we do the Ramsey experiment. So this is a kind of Ramsey fringes for the case where we just prepare this superposition and then do nothing, and then eventually apply another pulse and convert it back into a population. Or alternatively, what we could do, we could just send a weak pulse of light, and if we condition on one photon reflected from this cavity, what we see is that the phase of this Ramsey curve switches. It flips by pi. So this fringe here is contrast is somewhat reduced, and we understand why that is. It's actually because of finite probability of 2 photons being in the pulse. But what you see is that basically there are still a little bit of fringe, and most importantly, the phase of that is shifted by pi. But kind of what's most important is that one photon, for example, if we prepare the superposition, maximally coherent superposition initially, so if one photon hits this system, it all of a sudden changes the basically, the reflection probability, the output probability from nearly 0 to something like 70%. So that really corresponds to this one photon switch. And I think this is actually, very exciting because this really clearly demonstrates the ability to kind of do this photon switching at a single photon level, and opens up a lot of interesting possibilities for things like photon gates, atom-photon gates, photon QED detection, and so on. So we actually are quite excited about this, about this work, and we are now, already, carrying out the experiment involving atom pairs, and eventually we want to really scale things up, really prepare many of these, devices connected by the fibers. We also are now starting to explore this kind of many-body physics with strongly interacting photons. For example, this kind of physics of these bound states is actually very kind of interesting because it's far from equilibrium. It's really new kind of domain. So this, thing which still doesn't want to show this kind of the single photon switches and transistors, also might play a very important role in sort of all sorts of non-classical light generation. And there are some really, cool applications. For example, one could think about using this type of technique to create, for example, network of clocks, which would, form maybe something like a world clock. But maybe it's actually, I don't want to spend too much time talking about it. Okay, so at this point, I'd like to switch gears a little bit and talk about, something which is basically a different type of applications of this, quantum optics kind of nanoscience interface, which involves our effort to extend magnetic resonance imaging to nanoscale. So this effort, instead of cold atoms, it uses atom-like systems, which are basically localized defects in a solid state. And in particular, the one which should be most familiar here is the so-called nitrogen-vacancy color center in diamond. So it basically is a defect in which nitrogen atom, replaces a carbon atom in diamond lattice, and there is a missing atom vacancy nearby. So this actually is a system which in some ways behaves really like nature's own trapped atom or trapped ion. So the reason for that is that it actually is an optical emitter. It has an optical properties. It has a narrow emission line, kind of invisible. And what this allows one to do is to isolate this single atom-like defects with laser microscopy. So this is, for example, the picture of the diamond, which is, pretty pure, but not so pure. So what we see, these individual dots are actually individual optical emitters. And also what is important, in the ground state, this nitrogen vacancy has non-zero spin. So it actually spin triplet. One can rotate this spin with microwaves, and it has very long millisecond coherence time. So all of these things are known thanks to the early work in this field, which have been carried out by several remarkable individuals, including actually Phil Hammer, who is really one of the pioneers in this field. And certainly I learned about all of these things from Phil. So, but by now, one actually the kind of techniques for manipulation and fabrication of these NV centers advanced quite a bit. So, for example, what one can do now, one can create regular arrays or sort of quasi-regular arrays of these NV centers. The way how you do it, you first start with diamond, which you grow basically by CVD techniques, and you can grow a diamond which is extremely pristine, which is really completely free of defects. And then what you do, you basically bombard this diamond with nitrogen ions, and then you create this kind of arrays of this nitrogen vacancy center. So there is a lot of work now involving basically applications of these nitrogen vacancy centers to things like quantum information and quantum optics. But what I'll focus on today is really the use of these atom-like systems as nanoscale sensors. So, for example, one could think about using it as a sensor of magnetic field. And the idea is very simple. So this is basically an atom, frozen in the lattice. So you can shine light on it, and optically pump, by optical pumping, spin polarize this atom in one state. And then one can basically try to monitor the Larmor precession of this atom, and the rate of this Larmor precession will be proportional to local magnetic field. Right? That's a very simple idea. So that's, of course, the idea also for atomic magnetometers, which are among the most precise magnetometers, which you can now actually buy on the market. But there is something very special about this NV center, and that is the fact that this is really atomic scale impurity, which can be localized to basically nanometer scale volume. And what this means is that basically you can bring this kind of sensor very close to a target of interest. The target of interest can be single molecule or kind of single atom, single spin. So this basically allows you to do very sensitive imaging of magnetic field, where the sensitivity is basically determined by how coherent is the spin precession and very good resolution. So, and one of the kind of thing, this really opens the door for some very exciting potential applications. So one of those is to try to take basically conventional MRI and NMR techniques and scale them all the way down to like single, molecule or single spin level. But there is also some other, possibilities. For example, you can do nanoscale probing materials and surface. You can try to monitor elementary biochemical process. So this is actually by now a very exciting field. So I'll just give you kind of few, just a little bit of overview and talk about just one example of the recent work which we have done in this field. So some first applications which are emerging from this, one of them involves basically temperature sensing in living cells. So in this case, we use the sensitivity of an NV center not to magnetic field, but to temperature. What temperature does, it basically stretches a little bit diamond bonds, actually modifies also the response of the spin. And that way we can actually measure by taking diamond nanocrystals, putting them inside living cells. We can basically measure the temperature inside the living cells, and we can, for example, by heating the cells slightly, we could control viability. We could, for example, either keep the cell alive and happy or, kill it. So another also exciting opportunity is to use these NV centers for probing materials. And actually, we just have a paper coming out, I think it's this week or next. Yeah. Yeah. That's it. So these are like, this is, so here, this, basically was the idea to show the entire, so this is a cell, right? And, inside this cell, we have one nanocrystal, right? And so this nanocrystal kind of measures, in this case, this is not, this resolution is kind of limited, but, I will show you some, I have some nice pictures towards the future. So, yeah. That's right. So it's a temperature variation. So it's a temperature variation, delta T. Yes. No. So this is all done at room. So this is all done at, these experiments are done at room temperature, right? And what you do, you heat the cell by laser slightly and basically monitor, how temperature changes and whether the cell is happy or not, things like that. So another kind of example, which is maybe closer to a physics audience, so we can now probe basically magnetic noise in the proximity to the surfaces, for example, of conducting fields. And what we actually observed in a recent experiment, we basically observed a kind of transition, which is very simple, but still very kind of nice transition from basically a ballistic motion of electron in a kind of dirty metal to the basically to the ballistic transport. And the way how we did it is by just monitoring, measuring the Johnson noise in a kind of at a 10 nanometers or so away from the surface of the silver. Basically, what we can do is we can, show that, for example, Ohm's law is violated, provided you are working with the kind of very, clean single crystal silver material. But what I would really like to focus on today is actually our effort to really push kind of magnetic resonance imaging all the way down to the fundamental limit. And the approach which we are pursuing is actually, quite, simple conceptually. And of course, experimentally is very hard. So what we are doing is essentially we start with the diamond sample and then attach the target molecules of interest on the surface of the diamond sample. And essentially, using this NV center as a proximal sensor, what we are trying to do, we are trying to basically pick up the signals from nuclear spins in this single molecule. And we make use of the fact that these nuclear spins in the molecule presents with local magnetic fields with some frequencies which depends on this position on its environment. So basically, the big goal of this research, and I must say this, kind of idea has been like a holy grail in the field of NMR maybe for 50 years, but it completely seemed out of reach. But the big goal is to really map kind of positions of nuclear spins and chemical bonds with, resolution which is sub-nanometer, with really kind of atomic scale resolution. So I must say this really still looks like a very challenging goal, but what you will see now, we are actually making some progress towards realizing this goal. So these actually two people who are pushing this direction forward is Alex Soshkov as a postdoc and Igor Lovchinsky, and you see they are pretty happy, so things must be going quite well. Yeah, so it's actually this student is particularly remarkable individual because he was trained as a concert pianist, and I can tell you more. But okay, so there are two key ingredients which we need to really perform this kind of experiments. One is we need to manipulate our quantum sensor, our single electronic spin, which we try to use as a sensor of magnetic field. And in fact, we manipulate it in a very special way by putting some pulses of microwave radiation in such a way to basically synchronize the evolution of the spin, of the electronic spin, with the target nuclear spins. So in a given magnetic field, nuclear spins will be precessing at a frequency generally much lower than the precession of electronic spin. But what we do, we force the evolution of the electronic spin in such a way to kind of match the desired frequency. And that way, we can be specific to a certain type of nucleus. So, for example, by matching it to the frequency of, say, protons, we can detect protons. Or by matching frequency of carbon-13s, we can detect carbon-13. Exactly. There's a constant magnetic field. Yeah, nucleus spins rotate, right? Yeah, electron is also, and then in addition, we basically force electron spin to have, additional kind of low frequency, relatively low frequency. So, and then another thing which is also very important is that magnetic fields, for example, from point dipoles, they decay like 1 over a cube. So we need to bring our sensor very, very close. So that means that we typically will operate with an NV center, which is, 2, 3, 4 nanometers away from the surface, very close to the surface. Okay, but, basically once you sort of, have these two ingredients, this kind of nanoscale NMR is actually not that hard to observe. So what you can do, you can just take, for example, a drop of oil and just put it on the diamond. So oil contains protons, and it actually contains quite high density of protons, a known density of protons. And what you can do, you can just do this, perform these pulse sequences on this NV center, and then you see that, for a certain, kind of timing, so when the frequency of the pulses matches a lot more precession frequency of protons, you see immediately a signal, and the signal is quite big. So, and in fact, while these experiments, first have been done just maybe about 2 years ago, less than 2 years ago, this thing we now do routinely in the lab, and in fact, what we do is we use this kind of proton signal from oil to determine how close NV is to the surface. So we can determine the position of the NV centers with, like, basically with sub-nanometer precision. So, okay, so then, once we know where our NVs are sitting, so what we can do is we can start putting the molecules of interest, and actually we need to immobilize them somehow. So in order to do it, we developed together with my colleague in the chemistry department, Lukin Park, we developed a procedure where we can basically activate the diamond surface and attach then, proteins to the specific, covalently to diamond. And so then, for example, if we start with a very clean, very smooth diamond crystal, and then we put these molecules, so in this case, this is so-called ubiquitin protein. So in AFM, we can observe basically individual proteins sitting on diamond. Note now we can prepare this samples that actually density of proteins is sufficiently dilute. So the separation is something like 25 nanometers, and protein size is like 4 nanometers. So, and then, so we basically, can do it with different proteins. So in this case, we started with ubiquitin protein. Its structure is known, but it's kind of a model system. It's a modest size proteins with 100 spins. So in order to basically, have a clear signature from these proteins, we actually enriched it with carbon-13 and deuterium to distinguish from background, and then covalently attached it to the diamond, which was isotopically pure from carbon-12. And then in a case like this, we can immediately see basically both deuterium spectrum and carbon spectrum for certain NVs, not for all, but the ones which are close enough, and we can verify that these are the right type of species by just changing magnetic field. From data like this, from the strength of a signal, we can also find out where this protein is sitting. That's this white line relative to the NV center. That's an NMR spectrum, and it's obtained from single individual protein. Now, of course, what you would like to do is you would like to basically start, extracting some interesting chemical information. So, in fact, whilst you don't see this, maybe here, maybe a little bit, so you basically, so these lines are much broader than our spectral resolution. And the width of these lines depends, for example, of whether this the protein has deuterium or just has has just protons. So this deuterium, for example, has a large quadrupole shift, so we observe a large broadening due to quadrupole shift. So basically, when we just have protons, we observe the broadening of carbon-13 peak, which is just due to the carbon hydrogen interaction, carbohydrate hydrogen bond. So basically, what you see here is that these are clearly still some very simple baby steps, but what it really shows that we are now making first steps to do this NMR spectroscopy with some chemical specificity, and basically, these signals correspond to basically statistically polarized signals from just few nuclei. So let me just kind of, to conclude, let me just, take a step back and show you. So basically, where we are now and where we need to go. So in terms of NMR, conventional resolution of clinical NMR is something like 1 millimeter. So if you push the conventional techniques, you can go down to maybe 100 micrometers, and this corresponds to about 10 to 12 nuclei. And basically, with all sorts of advances, you can, with magnetic resonance microscopy, you can go down to maybe a few tens of nuclei. The data which I just showed you now basically correspond to few nanometer resolution, and maybe you can sense something like 100 to 1,000 of nuclei. But our goal is really to be here. And actually, we, have recently made some important progress towards this goal. So basically, the idea to reach these goals is to enlist some little help. And this little help, it turns out, can come from unpaired electron spins, which can exist on diamond surface. So usually, we don't like those kind of things because they cause decoherence of our NV centers. But at the same time, these kind of radicals on diamond surface can be very close to target nuclei. So the question is, can we use these radicals on diamond surface as a kind of reporters to sense local electric fields? So in other words, so imagine this is diamond, that you don't see that well. So you have an NV center, which sits maybe 4 nanometers below the surface. And so these are the surface electrons which exists on the surface. So we can use NV center to basically measure and control the state of these electrons. But now they can make via sensors. They can really measure what's going on within their local environment. So I will spare you details of this approach, but basically using NV centers, we can actually sense these surface spins. We use special pulse sequence. It's called double electron resonance sequence. And basically, we can actually verify that these impurities on the surface are actually spin 1/2. So they have resonance at the spin 1/2. Actually, moreover, what we can do by changing the magnetic field and using the fact that the dipole-dipole interaction is anisotropic, we can actually localize where these reporters, where these spins are sitting relative to NV center. And we can localize them with very high resolution. So the most proximal one, we, for example, can localize down to sub-nanometer resolution. Exactly. So this basically NV center interacts with, exactly, and it's a very good, so because it's still few nanometer separation. So and these guys exist on the surface. As you see, the separation between them is also few nanometers, right? And basically, you use NV centers to kind of find out to sense signals from them. Now, remarkably, these guys, even though they are radicals living on the surface, they are remarkably stable. And they are also remarkably coherent. So, for example, what one can do is one can drive Rabi oscillation on the surface spins. And what we see, these Rabi oscillations exist for like a microsecond. These are all room temperature ambient conditions experiments. That's it. So these are electrons. So now on the surface, these are electron spins. This spin 1/2. So these guys are electron spins, right? So they are have much larger. Exactly, these are electron spins. So they are easily separated from everything else. We can discuss why this decay occurs, and that's actually an interesting question. But moreover, you can also measure the T1 time, the spin orientation time, and actually it's even longer. It's something like 40 microseconds. So that means that with some proper control, probably we can even extend this. So this thing most likely depends on on dipole-dipole interaction of this surface nuclei. So basically, now kind of what we sort of the idea which we try to pursue. Now let's use these guys, these spins on the surface as sensors. Right? This is just our readout window, and those guys will sense what's going on around. And what we'll basically do this, we apply these pulses, the same as we applied for NV center, on this spin 1/2 electrons, and just use our NV center as a readout. So, and in this case, we did not put anything on the top. So all we do is just probe protons on the surface. There are always protons, even on a free surface, for example, due to things like hydroxyl groups. And basically, we readily observe that there are protons on the surface that you interact with these reporters. So this is kind of a reporter spin state, and what it shows is some kind of modulation of the reporter spin, basically coherence. And so this modulation, it turns out, is exactly occurs at the Larmor frequency of the protons for most of the values of magnetic field, but not for all. So it turns out there are some kind of special resonances where some very special frequencies all of a sudden appear. Actually, it turns out that at these resonances, basically these reporter spins evolve coherently, and actually the origin of this is coherent coupling of these reporter spins, two nuclear spins, two protons on the surface. So this coupling becomes resonance when magnetic field basically compensates the hyperfine interaction frequency, basically that allows us to essentially find out what's the hyperfine interaction is between these reporter spins and nuclei on the surface. So from there, what we can do, of course, we can immediately try to reconstruct where these nuclear spins are located relative to these reporters. And this can be done with actually quite amazing resolution. So here is the position of a reporter, and relative to it is the position of two protons. So if you assumed that these protons are point dipoles, you can really localize them extremely well. So this circle, however, indicates that, since we don't know exactly the chemistry, so there might be some contact interaction. So this is essentially, if you want, the position, the size of the wave function of this proton. Okay, so I think I've really used up my time, but I hope this kind of shows you that the techniques which we are developing really push us towards this fundamental limit of magnetic resonance imaging. So we have, of course, big plans, how we can now apply this reporter kind of networks to, for example, sense signals from single proteins. We are adding gradients. We are learning how to control and polarize these reporters. At the end, what we can really probe, we're already starting to do it. We can probe really coherent dynamics and control of spins in these single proteins. And I think there are some important applications which, for example, we have in mind. One of them includes studies of conformational changes in structure of membrane proteins. So these are the proteins which cannot be crystallized, and the structure, most of them, the structure is not known. Okay, so I hope I gave you a little bit of a feeling, about this new interface between quantum optics and nanoscience. So on one hand, it really provides us with new platforms to study kind of quantum optical phenomena and maybe pursue these ideas of quantum information science. But at the same time, these quantum techniques really allow us to create new generation of sensors, where we basically use the fragility of atomic coherence and the fact that these sensors are small to really do some kind of interesting things. And so with that, I would like to thank you for your attention.